Click for IB Biology. Here we're looking once more at the simple diagram of our membrane, the lipid bilayer structure. And we're looking at how it's acting as a barrier to exchange, regulating what goes in and what goes out. And we'll begin our analysis of that by looking at the process of simple diffusion. And of course, students will be all too familiar with the idea of substances moving from a region of their high concentration to a region of their low concentration. But this is a special case of simple diffusion that we're going to be looking at first. There's a blank PDF of this presentation down in the description. There you'll also find a quiz of 10 questions with answers and perhaps some other things down there for you to benefit and add to your learning. So let's get into this simple diffusion, the movement of molecules across the cell membrane from a high concentration to a low. Let's remind ourselves of the model that we're using. First of all, remember that the model that I've been explaining in the previous one was that of a sphere. We're thinking of a spherical cell where the phospholipids are organized into two layers, with the phosphate heads on the outside and the phosphate heads on the inside. And this is because of the presence of water on both the outside and the inside of the cell. So the extracellular tissue fluid would be water and the cytoplasm would be water. But what we find is that the bilayer of fatty acid tails, which are hydrophobic, remember, they are nonpolar, they will not allow the movement of charged particles like ions across the membrane. So sodium ions, potassium ions, calcium ions, they cannot freely move backwards and forwards across the cell membrane because they are repelled by the hydrophobic layer in the center. So the hydrophobic fatty acid tails can be thought of as a barrier, and it prevents the movement of large polar molecules, and they can't pass through. They are repelled by the hydrophobic layer. So to simple diffusion. Simple diffusion is limited to almost to two extremes of molecules. We've got the very small molecules, very, very small molecules like oxygen and carbon dioxide at one extreme. And these molecules are so small that they are not subject to the repulsion of the fatty acid tail layer. This bilayer of fatty acids does not prevent the movement of oxygen and carbon dioxide across the cell membrane. So this is two very important gases, of course, from respiration, but the cell membrane is not a barrier to the movement of those gases, and therefore they will move from a high concentration, typically in oxygen high concentration outside the cell, will move to a low concentration region inside the cell. Carbon dioxide will move from a high concentration inside the cell to a low concentration outside of the cell. Now, I said there was an extreme, and at the opposite extreme of the molecules which can move through the cell membrane are very, very large molecules called steroid molecules, and many of which are hormones. So these large steroidal molecules, the reason they can pass through is because they themselves are nonpolar, and so the cell membrane fatty acid tails do not act as a barrier to the movement of our steroidal molecules. And so this is important in understanding the action of molecules such as hormones in cell signaling, that they can simply pass straight through the cell membrane and into the interior of the cell. But that would have to be a steroidal hormone. Some hormones are protein and they cannot pass through the membrane in this manner. Simple diffusion, the passage of the tiny molecules such as oxygen and carbon dioxide from a high to low concentration and the big steroidal molecules. Simple diffusion. Here's the link to the next video in our study, to the channel, which I'd be grateful if you could subscribe to, and there's the playlist with all the other ones on cell membrane, all the other videos.